Hello, welcome to another video, and here I want to share this original article with the title Cross-Platform Validation of Neurotransmitter Release Impairments in Schizophrenia Patients Derived NRX and 1 mutant neurons. So heterozygous NRXN1 deletions predispose to schizophrenia and other neurodevelopmental disorders. Engineered heterozygous deletions impair neurotransmitter release in human neurons, suggesting a synaptic pathophysiological mechanism. In a multi-center effort to test the generality and robustness of this pivotal observation, we used at two laboratories independent analysis of patient-derived and newly engineered human neurons with heterozygous NRXN deletions. So NRXN1 deletions exhibited the same measured decrease in neurotransmitter release and an increase in protein and as key protein as engineered human neurons with NRXN1 deletions. Strikingly, engineered mouse deficient neurons derived by the same method displayed no such phenotype, suggesting a human-specific role for NRXN1. Thus, heterozygous NRXN1 deletions robustly impair synaptic function in human neurons, enabling future drug discovery efforts. So they mentioned the role of deletions in schizophrenia patients, and they use also animal study, so that I think they have like three sample, like two human sample and two sub-samples of humans and the animals here. So let's read the abstract. So heterozygous NRXN1 deletions constitute the most prevalent currently known single gene mutation associated with schizophrenia, and additionally predispose to multiple other neurodevelopmental disorders. Engineered heterozygous NRXN1 deletions impaired neurotransmitter release in human neurons, suggesting a synaptic pathophysiological mechanism. Utilizing this observation for drug discovery, however, requires confidence in its robustness and validity. So, using neurons transdifferentiated from induced pluripotent stem cells that were derived from schizophrenia patients carrying heterozygous NRXN1 deletions, we observed the same synaptic impairment. This impairment manifested as a large decrease in spontaneous synaptic events, in evoked synaptic responses, and in synaptic paired pulse depression. So they tried using mouse neurons generated from embryonic stem cells by the same method, as human neurons didn't exhibit impaired neurotransmitter release, suggesting a human-specific phenotype. So the deletions produced a reproducible increase in the levels of CASK and intracellular NRXN1 binding protein and were associated with characteristic gene expression changes. Thus, heterozygous NRXN1 deletions robustly impair synaptic function in human neurons regardless of genetic background, enabling future drug discovery efforts. So that seems to be a very interesting study, including different perspectives, different approaches to analyze the hypothesis, and there are many authors affiliated with this study. I think there are at least 15 people here, and thanks for watching this video, and I wish you have a beautiful day. Bye bye.